Up next, we have Guilherme Caeiro-Dias with the Department of Biology and Museum of Southwestern Biology at the University of New Mexico. And the presentation is the next generation of Rio Grande Silvery Minnow genetic monitoring. So I will go ahead and turn it over to Guilherme to share your screen.
So, thank you very much for attending to my talk. My talk is entitled "The Next Generation of Rio Grande Silvery Minnow Genetic Monitoring." My name is Guilherme Caeiro-Dias. I'm a biologist and postdoc researcher in Turner Lab. I'm working with Thomas Turner and Megan Osborne, and my main research consists in the use of bioinformatic tools to generate genomic data for population genomic analysis and applied for conservation, essentially. So before I show you some of our results, I will just give you a general idea about how population demography can affect genetic diversity. And so as we know, population characteristics as rate of change, abundance, or distribution change over time. And quite often these changes in natural populations are accompanied by changes in the genetic diversity. So as an example, I have here this plot that shows the changes in the population size over time. And each circle represents one individual, and different colors represents a variant of for example, a gene, a particular gene. And we see that when the population rapidly decreases, the rarest variant was lost. And this is something that quite often happens in a bottleneck event. Another way that population may lose genetic diversity is through genetic drift, which is essentially random changes in the frequency of these variants. And because they are rare variants, because they are rare, they are more susceptible to these random effects. So as you can see, in the end, the genetic diversity in the population is quite different from the beginning. There are other factors that influence genetic diversity, like population evolutionary history or natural selection, other characteristics of the species. But here we will focus on the demographic factors. And the Rio Grande silver minnow is particularly susceptible to demographic fluctuations, mostly due to a highly variable environment, but also because of its relatively small population nowadays. So this is why it's important to keep track of these changes and monitoring these changes, particularly for management decisions. And the Rio Grande silver minnow has been monitored during the last decades. So in 1994, I have here a timeline that showed the three main events, or three important events for the Rio Grande silver minnow conservation. This species was listed in the Endangered Species Act in 1994, and the genetic monitoring started in 1999. Since then, samples were collected every year for genetic monitoring, and in 2002 started the augmentation program. So just let me say that the genetic monitoring started in 1999 has been done using a type of genetic markers called microsatellites. Here you can also see these fish bones that represent years where the population suffered major declines. And also on top of some ears, you can see those tubes that represent the samples that were used in this study. So the main results from previous genetic monitoring, or the genetic monitoring using microsatellites, was that even with the high fluctuation in the population size of the silvery minnow that is represented here in this left plot, This is actually the evidence since 1993. Despite this fluctuation, the genetic diversity has been maintained quite stable across time. Here we have three common measures of genetic diversity. Heterozygosity, that essentially measures the proportion of markers in individuals that have two distant variants of the same marker. Here we have the inbreeding coefficient, which essentially measures how related the individuals are in the population, and the allelic richness is the number of genetic variants in the population, and here corrected for sample size. So you can see here some fluctuation in the inbreeding coefficient, but considering the y-axis varies between 0.1, 0.25, this is a quite small variation. So what did we do different from what has been done so far? We used the genomic approach called red sequencing to get, which allowed us to identify almost 3,000 genetic markers containing more than 5,300 variable nucleotides, which we call SNPs. And these genetic markers are scattered across the genome, and then they should be a good proxy to estimate diversity at the genomic level, and also to detect small changes in the genomic composition, even in the population, even within few generations. So first we measured the same measurements as for microsatellites, measurements of genetic diversity. And we have the heterozygosity, allelic richness, and inbreeding coefficient. And we see that the patterns across time are essentially very similar. So the conclusion is similar to microsatellites. We have a very, the diversity has been maintained stable across time. What we additionally know to microsatellites is now that we can identify how the diversity varies across the genome. And we see that, for example, with heterozygosity, we have a large group of markers that show small heterozygosity. Oops, I'm sorry. But also we have a large group of markers that show higher heterozygosity, and some markers with intermediate values. This is more or less what happens for the allelic richness. And for the inbreeding coefficient, it's a bit different. All the values are more or less around 0, which is expected when the reproductive population, when individuals in a reproductive population are not related to each other, which when population is large enough, that's what happens. So the only exception is this 2 years here, 2009 and 2010, where we see a bit in an increase of the values of the inbreeding coefficient. So we have more markers with larger values than previous years, and also then the next years. So our question was, what happened during those 2 years for inbreeding to increase? So we measured, we have genetic estimations of the reproductive population, and those measurements were quite low. Probably they are underestimated, especially when we compared with the number of individuals that were estimated, with evidence that were estimated for those years, particularly in 2009, it's quite high. So these values are quite contradictory. But somehow the population, the individuals that reproduced in these 2 years were more related to each other than previous years, which we don't have still a definitive explanation for this. But at the same time, during these 2 years, also the number of captive reared fish released was very, very low. Which contrasts with the next years where those numbers increased. And so this may explain why in the next years, the inbreeding coefficient decreases again. So probably the population was, the reproductive population was more related to each other in those 2 years, and introducing more individuals in the river created a more diverse system. So the individuals were more unrelated between them, and this decreased the inbreeding coefficient in the next years. So next, we estimated the FST, which is a statistic that measures the changes in frequency of genetic variance between years. And what we can see is that most of the markers didn't change across time. So the values are very close to 0, but then we have a small number of markers with small changes in allelic frequencies. And our question is, did these small changes occur throughout time, or did they happen in a specific point in time? So to answer this, we use the discriminant analysis of principal components. This analysis essentially describes the diversity between predefined groups, and in our case, the groups are each of the years. And we see that before 2012, most of the populations, most of the years largely overlap. We have here a group that has a smaller overlap, but this variation is mostly across the y-axis, while the x-axis explains larger variance. And so these 3 groups here that correspond to the last 3 years of our sampling show a higher deviation from all the previous populations for all the previous years. So what happened between 2012 and 2015 to explain these differences? So during those years, the population suffered a major decline, and at the same time, the population was supplemented with a large number of individuals. So it means that essentially during those 3 years, the natural population, the population in the river, was essentially constituted by fish that were released from captivity. And so these small changes in the genomic composition may be related with a small genetic drift effect associated with the supplementation. So here our main findings are that the genomic diversity, as shown before, was maintained across time. Also, the increase of inbreeding levels was reverted after supplementation, and we detected only a minor genomic shifts in genomic composition shifts even after a major bottleneck. And this shows how important is the augmentation program to keep the genetic health of the Rio Grande silver minnow. I didn't point here, let me just go back, that 2018 is the year that deviates most from all the other years, including even comparing with 2015 and 17. And so it may suggest that these shifts are still ongoing, but we need samples from the next years to confirm this. So what we're going to do next, and this is what we are working on now, we will select a subset of markers that are suitable for genetic monitoring. And you may ask why we are not using all the markers that we have now. So the technique that we use, the red sequencing, the replicability of this technique is very high, but still it's almost impossible to get every time the 100% of the same loci, every time that we repeat this, the same markers. So, we need to make sure that we are always monitoring the same genomic regions. And also because of costs, it's cheaper to sequence less markers than more markers. So we will use a technique called GT sequencing, which is similar to red sequencing, and it allows to amplify and sequencing specific markers in a large number of individuals. But the main restriction is that it is efficient only for a smaller number of markers, about a maximum of 500 markers. And this is, we already have preliminary results, and we should be able to have a final panel soon. And thank you very much for to all these people that helped somehow in this work, and without them, it wouldn't be possible to accomplish this. And thank you very much for listening.